Hey guys, welcome back. Today we're going to be covering chapter 20, which covers phylogeny from Campbell's Biology in Focus. Okay, so it starts off with this legless lizard. You look at this picture and you're like, that's a snake. It's actually not a snake, it's called a legless lizard. So the beginning of this is talking about this lizard because it is on the first page of the chapter. Okay, so we're going to look at investigating the evolutionary history of life. We started off with descent with modification in our last lecture. So this lizard that you just saw a picture of, it's considered a legless lizard. And these lizards and snakes actually evolved from different lineages of lizards. It wasn't like snakes came later than lizards. It's just two different lizards that have evolved separately to have a legless lizard and snakes. So these legless lizards have evolved independently in several different groups through adaptation to similar environments. So remember that we talked about that previously. Sometimes when organisms are faced with the same challenges in different locations, that they end up having similar structures because they're trying to, adapt to their particular environment. So here's kind of what that would look like. This is like a small phylogenetic tree. This is more of like a clade, which is just like a group. Okay. But you can see here that you have ancestral lizards with limbs. So very, very old lizards, ancestral lizards that have limbs, so they have legs. And then you can see each of these branch points. The first one to break off was geckos. So geckos are like considered to be, quote, old lizards. And then you have another branch point over here that split immediately to snakes and then everything else that had legs. So snakes came first here. So the snakes developed this no limb trait. That's what this is. It's a trait that they developed. Okay. And then moving on to the other side of this little bracket, we have our legged organisms. And then independently, this eastern glass lizard developed no limbs. It must have had a similar environment as our snakes here. So they evolved to have similar characteristics. Okay, so phylogeny is what this chapter focuses on. So phylogeny is just the evolutionary history of a species or a group of related species. So this is kind of what it's looking like. It's a way to diagram relationships between different organisms. So the discipline of systematics actually helps to classify organisms, and it's going to help to determine their evolutionary relationships, because that's all that we're doing here. So like we have different ways of naming organisms. We have different ways of displaying evolutionary relatedness in phylogenetic trees, which is what we're going to be looking at in this chapter. So phylogenies show evolutionary relationships. That is the point. So taxonomy is the ordered division and naming of these organisms. So when we have these phylogenies that show the evolutionary relationships, we're then able to divide the organisms up further and then apply names to them. And you've probably heard of binomial nomenclature. Carolus Linnaeus is the one that kind of came up with this naming system for taxonomy. So he published a system of taxonomy based on resemblances, so the way that things looked. So we know that their evolutionary relatedness is not the same as the way that they looked, their resemblance. Because as we just learned from these glass lizards, they're similar looking to snakes. So their name is probably more similar to the scientific name of a snake than they are evolutionarily related, if that makes sense. So sometimes there's a divergence there. So there's two key features of the naming system that still remain useful today, even though now we're relying more on like the facts of evolution, evolutionary history. There's a two-part name for species and hierarchical classification. So you know that binomial nomenclature means two naming system. Okay, so it's going to be the genus and the species for an organism. And so the classification system is where we get those genus and species. It's the two most specific. So each organism on the planet has one genus, one species, and that is its kind of like identification. So for humans, it is Homo sapien, our genus and our species. So that's what this is going to talk about here. It's a two-part scientific name based on the species. Okay, so it's called binomial because it has two names. It is the genus and the species. So the first part of the name is genus. The genus is capitalized. They're all italicized, but the genus is capitalized, the species is not. And both parts of the name, both parts together name the species, not the specific organism alone. Okay, so we all know Dear King Philip came over for good spaghetti, right? That's the classification system. That's the like little acronym to help you remember it. So Linnaeus introduced a system of grouping species in increasing broad categories. So we kind of started off with like, oh, these are all animals. Great. Animalia, animals, if it's an animal. But there's a lot of differences there. So then you're able to further divide that into another subset, and then those organisms into another subset. So that's kind of how we got this hierarchical classification system. So the taxonomic groups from narrow to broad are the species and the genus. So those are the most specific, because genus and species makes up our binomial nomenclature scientific name. So from the most narrow, one organism, to the most broad, it goes species, genus, family, order, class, phylum, kingdom, and domain. So the domain is the most broad category. There are 3 domains of life. Everything that has ever been alive is classified into these 3 groups. Well, it's not very specific, right? But a genus and the species are extremely specific. So a domain is extremely broad. A kingdom is a little bit, there's 3 domains, there's 6 kingdoms. There's like 20 something phyla, right? So then it increases in groups as you go down towards species, because there's a whole bunch of species, right? So it's getting more and more and more and more specific as you go from domain, kingdom, phylum, class, order, family, genus, and species. Species is the most specific. This is how that looks. Okay, so the animal here is like a leopard, okay? So if we start down at the bottom, that's the most broad category, right? There are 3 domains of life. This is domain eukarya. Why is it considered domain eukarya? It's an animal. Animals are made of animal cells. An animal cell is an example of a eukaryote. Eukarya. It has eukaryotic cells. So then that's the most broad. Next, we're going to move up to kingdom. Kingdom, animalia. That makes sense because this leopard is an animal. Are we very specific? No, because you could also have a fish in here, because a fish is also an animal, and we're not trying to describe a fish. Okay, then we're going to go into phylum chordata. Okay, this is something that has a spinal cord. We're going to go into class, mammalia. This is a mammal, so something that has mammary glands, gives live birth, and produces milk for its offspring. Carnivora is the order, something that is a carnivore. It eats meat. Okay, Felidae is the family. I believe that these are cats that don't roar. I believe that's what Felidae is. Genus is Panthera, and then species is Pardus. So the binomial nomenclature for this organism is Panthera pardus. Okay, so the genus and species are the most specific because that's describing this one organism. Now, lions are also large cats, but they roar. So they're going to have some slight differences here. They're still going to be in the same domain, kingdom, phylum, class, order, but then they might start to split off a little bit. They're still related, but not in the same genus and species because they are not the same organism. Okay, so a taxonomic unit at any level of hierarchy is called a taxon. If you have multiple, it's called taxa. So the broader taxa, multiple groups of organisms, are not comparable between lineages. So for example, an order of snails is less genetic diverse, has less genetic diversity than an order of mammals, because there are a bunch of mammals, whereas there's a few, of that same order of snails. It's just you can't be like, oh, if you're comparing one phylum to another, that doesn't mean that they have the same diversity, the same number of organisms that are present at that level of classification, if that makes sense. It's not like you're comparing apples to apples there. It's not like, oh, if there's 32 organisms that are in this one phyla, there must be 32 organisms in this other one. That's not true. It just depends on, which organisms are proven to be most related, and then they're grouped together. Okay, so linking classification and phylogeny. So I said that this chapter is about phylogeny, which are those like pictures. It's like a diagram, a nice little evolutionary history, whereas classification is more like based on resemblances. So systematicists developed evolutionary relationships in branching phylogenetic trees. So they use some of the information from classic classification practices, but then also with more of our modern like DNA research in order to discover evolutionary relatedness between organisms to develop our phylogenetic trees. So this is an example of a phylogenetic tree. This one is dealing with these mammals. So you have a leopard, an American badger, European otter, a coyote, and a gray wolf. So it starts off at the order. So we know that we had Dear King Philip came over order for good spaghetti. That's what that order, family, genus, and species. Dear King Philip came over, that's where we're starting, order, for, family, good, genus, spaghetti, species. Okay, so we're starting with carnivora. So these are all carnivorous organisms. Then we're going to go into family. So from carnivora, you have a split. This is called a branch. Okay, these little points are also called nodes. It's also representing the most common ancestor. So whatever was here was a common ancestor for everything else. Okay, but we have a bracket here, and this branch developed into Felidae. And this branch ended up splitting again. I'm not sure how to say these words, but these are other families for organisms that developed later, because obviously these two organisms are going to be much more related than these two organisms, which we know are related to our domestic dogs, right? So carnivora had a split, Felidae, and then this other branch down here that immediately hit another node. There was another deviation, which is fine, happens a lot in evolution, right? And then we have another split here to lead us to our American badger and our European otter. So these two being on the same branch here, they are sister taxa and very much related to each other. Same thing with the coyote and the gray wolf. Because they immediately share this node, they are very related to each other, whereas the coyote and the European otter are this branch point, and this branch point. They are 3 branches away from each other, so they're not very closely related, but they are still in the same order. So there is a similarity between them, but then we have separated them into their own genus and species, family, genus, and species based on their characteristics and evolutionary history here. Okay, so the one that's kind of the outlier is the Felidae. It's going to be the least related to the others because it kind of diverged first. And we're going to talk more about this. It'll make more sense. Okay, so Linnaean classification and phylogeny can differ from each other. So that's what I was saying. The classification is based on resemblances, and phylogeny is based more so on actual evidence of evolutionary relatedness. So systematicists have proposed that classification can be based entirely on evolutionary relationships. That's kind of more of like an opinion because it was originally developed based on resemblances, which if organisms resemble each other, sometimes it is true that they developed in the same kind of environment and are very closely evolutionary related. Is that true 100% of the time? No, of course not. Science likes to have exceptions. So phylogenetic tree represents a hypothesis. It's a guess because we don't actually know, right? We just have information that has led us to believe this to be true. So it's a hypothesis. This is an educated guess about evolutionary relatedness and relationships. So each branch point represents the divergence of 2 taxa, so 2 groups from a common ancestor. So one organism that was the ancestor to the 2 that have diverged. Sister taxa are groups that share an immediate common ancestor, which I pointed out on the other picture, but I will point them out again in just a minute. You have a rooted tree that includes a branch that represents the most recent common ancestor of all the taxa in the tree. You have a basal taxon, which is one group. It's kind of like removed from the rest that diverges early on. So that would have been like the Felidae in the last example. An early history of a group and originates near the common ancestor of the entire group. And then you have a polytomy, which is a branch where you have more than just 2 divergent organisms coming off of it. So we're going to look at a picture to kind of point these things out. Okay, so here's a phylogenetic tree. You have this is kind of like what I think of like the beginning of time and then like now, right? So you're kind of like moving through evolutionary history and you're also showing relatedness here. So you have the ancestral lineage here and you have a branch point. So this is a common ancestor. So this branch point represents a common ancestor to taxa A through G. So this organism is where all of these came from, right here, whatever existed here. We have a branch point, okay? This first one to break off, taxon G, so group G, whatever this organism is considered the basal taxon. It's the very first thing to split off. It splits off in early evolutionary history. Then if you look at the top part of this bracket, you have another branch point, which is where you have lineages diverging, right? So you have a bracket here. Now you have your common ancestor 3 and common ancestor 5, right? With common ancestor 3, you kind of have like another, quote, semi-basal group that's splitting off here. Taxon A. And then you have another side of the bracket that has yet another node where you have common ancestor 4 that branches off into taxon B and C. These are considered sister taxa because they share an immediate common ancestor. Okay, so over here with our lineage that diverged from common ancestor 5, this is a branch point that's forming a polytomy, an unresolved pattern of divergence. So just unresolved pattern just means out of these, we don't know which one came first. We just know that these 3 organisms share a common ancestor, and it's whatever was at position 5. Okay, so up here, I would also like to point that out for like common ancestor 4 taxons, the taxa B and C. If you were to flip them and put C and B, it's like you're spinning on this little section right here. Like if you were to just rotate this section, you can do that, and that's still correct because this doesn't indicate which one came first. You don't know that, right? Here, this one came first, and then we have a split, and then we have a split, and then we have another split, and we have a split here, and we have a split here. We know that this one came first, but here, you're sharing a node. You don't know which one of these broke off first, if that makes sense. So these have rotational similarities. Okay, here, same thing. This is the polytomy, which means that we have more than 2. Now we have 3 that are branching off from a common ancestor. And the unresolved pattern just means that you don't know which one came first, which is fine. It's just that they all came around after whatever number 5 represents. That's how you read one of these phylogenetic trees. Okay, so what we can and cannot learn from phylogenetic trees. So phylogenetic trees show patterns of descent, not phenotypic similarity. So this is not because 2 organisms look the same. That is their phenotype. Okay? They are showing you descent. It's showing you descent with modification. This is evolutionary history. And we know that not because 2 animals might look alike, but that doesn't mean that they came from the same place. And we learned that with the flying squirrels in the last lecture. So phylogenetic trees do not generally indicate when a species evolved or how much change occurred in the lineage, which is also something really important to explain. So just because this line is really long, it doesn't mean that there was a whole lot of change occurring here. Sometimes that's a common misconception. Like this line is very short. So this was obviously, a smaller change. Well, that's not necessarily true. It's just showing you we have a sheet of paper with so much room, and we're trying to make this as easy to read as possible. So this is going to be a shorter branch because you have all of this stuff that came before, right? So this divergence here of taxon G does not mean, oh, look how much time has passed. This line is not equivalent to time, so you can't think about it like that. But that is a very common misconception, okay? And it should not be assumed that a taxon evolved from the taxon next to it. Oops. So that just means B and C did not necessarily evolve from each other. It's descent with modification. They shared a common ancestor at some point in time. Or taxon C and taxon D do not necessarily evolve one from the other because, look, there's a whole bunch of taxon D's most common ancestor is 5. Taxon C's is 4. And then before that, it's 3. And then from before that, it's 2. They don't really have a common ancestor in recent evolutionary history. So you kind of have to go through and look at the entire diagram here. You can't just be like, oh, C and D are close together right here. Well, yeah, their names are, but are they actually close together? No, not at all. The sister taxa have the most relatedness. Okay, so applying our phylogeny. So we kind of know how to read a phylogenetic tree. We kind of know what it's representing. We know miscommunications that the lines do not represent time. Okay, so phylogeny provides important information about similar characteristics and closely related species. Like I said, our sister taxa are going to be the closest related. Phylogenetic trees based on DNA sequences can be used to infer species identities. So DNA sequences are the best evidence of evolution because if you have a similar DNA structure, you have similar genes, you have similar traits in your genetics, that means that you had to come from somewhere similar, a common ancestor. So for example, a phylogeny was used to identify the species of whale from which whale meat originated. So you can use them to, the ones that are based on DNA, specifically, you can kind of infer other species, because if you know some of their genetic code and then you have another piece of genetic code that you're like, well, where would this fit, right? You're able to better identify different species that way, because you have this comparison. It's kind of like a giant comparison chart in order to like kind of relay information back to. So this is kind of like an image of what they're talking about here, how you have unknown species, but based on phylogeny, you're able to kind of be like, okay, well, it's kind of close related to, like 9 is most closely related to the mink whales, right? And then the humpback whale is most closely genetically related to the unknown number B1 or 1B, right? So you're kind of able to go through when you're dealing with just phylogenies that are built off of DNA and say this genome sequence is similar to this genome sequence, and they express the same genes, they make the same proteins, right? That's how they're able to make something like this work out. So phylogenies are inferred from morphological and molecular data. So sometimes there's different types of phylogenies, right? So we're seeing that this is yet another one. So to infer phylogeny, systematicists gather information about morphology. So the way that it looks, like its shapes, its overall shapes, because we talked about homologous and analogous structures in the last lecture. This is the morphology of it. It's not just like, oh, it's brown. They're all brown, so they got to be related. Like, that's not quite right. We're talking about like how the human arm has similar bone structure to that of a whale fin. Somewhere they had to have a common ancestor. These morphologies, the structures, okay? Genes, which we get from DNA, which is the best evidence of evolution, and the biochemistry of these living organisms, which we've also touched on in the last lecture. So then similarities are used to infer the phylogenies, and they must result from shared ancestry, right? If these two organisms display the same traits, they've got to come from the same place, right? That just makes sense. Okay, so morphological and molecular homologies. So phenotypic and genetic similarities due to shared ancestry are called homologies. So notice that it says due to shared ancestry. So if you have a common ancestor and you have similarities, whether they're phenotypic or genetic similarities, that's evidence of a homology, which is one of our pieces of evidence for evolution. So organisms with similar morphologies against structures or DNA sequences are likely to be more closely related than organisms with different structures or different DNA sequences. Well, that makes sense. If you have the same basic bone structure, even if the bones are slightly different sizes, then you're probably related. Whereas if you have a completely different bone structure, I would say you're not very closely related. And that's exactly what our morphological and molecular homologies help us to determine. So sorting homology from analogy. So homology is things that are similar. Analogy is when they sometimes have similar structure but different purposes. Okay, so when constructing a phylogeny, systematicists need to distinguish whether a similarity is the result of homology or analogy. So homology is similarity due to shared ancestry, which means that they have a common ancestor, that they descended from the same organism. Whereas analogy is similarity due to convergent evolution. So think about, like, I'm going to talk about analogy. Anything that has a wing, a bird, a bat, a butterfly. These things all have wings and they all fly, yes? Okay, cool. That's not showing you shared ancestry. They have analogous structures. It's the same function, but it's a different kind of structure. What is the function? The function is to fly. That's why these organisms have wings. Butterfly flies. A bird flies. A bat flies. Correct. They have a wing and its job is to fly. But are they all built the same? Is a butterfly wing made the same as a bird wing? Well, think about it. A bird wing has bones that are very porous because they're very light. It has, veins. It has skin. It has feathers. Think about a butterfly wing. It's made of mostly like chitin, right? Like it rubs off in your hands if you accidentally touch a butterfly's wing. Think about a bat. A bat is a mammal. It has skin. It is a warm-blooded organism. It has little bones and these long finger bones, these long phalanges, and a bunch of skin flaps between all of them that allow it to fly. Now, are these structures the same? No, absolutely not. They're not the same. Is the wing, the purpose of it, the same? Yes, its job is to fly. So that is what an analogy would be, an analogous structure. Okay, so convergent evolution occurs when similar environmental pressures and natural selection produce similar or analogous adaptations in organisms from different evolutionary lineages. Like I just said, a butterfly, a bat, a bird. They do not share an immediate common ancestor. We have an insect, we have warm-blooded and cold-blooded organisms, right? A mammal is not going to be in the same group as an insect. They are both going to be, very, very, very distantly related, sure. But these wings developed because of environmental pressures and natural selection to their environments. These are analogous structures. Their job is to fly, but they are not composed of the same structures. They are showing you that they have different evolutionary lineages. And that's like something that's kind of hard to wrap your mind around, but I hope that the wings made it pretty relatable. This is also true of these little moles, that they have similarities, but they're also an example of convergent evolution from the textbook. Okay, cool. This is talking about wings. Look at this. Okay, so bat and bird wings are homologous as forelimbs, okay, but analogous as functional wings. So it's saying that the structure of the bones is similar as forelimbs, but analogous as functional wings, because they did not develop wings from the common ancestor. They developed their arms, their forelimbs, from a common ancestor, but not for the function of flying. Okay, so analogous structures or molecular sequences that evolved independently are also sometimes called homologies. Okay, so this is when we have a homology that can be distinguished from an analogy by comparing the fossil evidence and the degree of complexity. So we refer back to fossils, we refer back to the molecular, the DNA, right? We refer back to all of these things to kind of prove whether we're dealing with analogy or homology here. Okay, and the more complex two similar structures are, the more likely it is that they are homologous. Because if it's very complex, it's not going to be something that is conserved unless it came from the same place, right? A very complex structure, if it's similar in two different organisms, they probably came from the same place, because to have that complexity develop because of two different environments that had similar environmental pressures is just kind of like a 1 in a million sort of chance. Whereas the more complex something is and it's conserved among a lineage, that would make sense because it's coming from the same common ancestor. So it's just slight modifications on that one particular complex structure. Okay, we're going to be looking at evaluating molecular homologies. So molecular homologies are determined based on the degree of similarity and nucleotide sequence between taxa. So what's a nucleotide sequence? That's the A, T, G, and C in the DNA. Okay, whatever combination of A, T, G, and C that they have. The more similarities we have, the closer we can tell we have molecular homologies. So systematicists use computer programs when analyzing comparable DNA segments from different organisms. It's called a BLAST database. We actually have a bunch of the genomes already sequenced of different organisms. We have their genus and species on file with all of the genomes that's related to that. And then you can compare that to another organism and it'll tell you how many similarities they have, because a computer can go through and count millions of bases in seconds, whereas it would take a human forever to do that. So here's kind of like an overall example of showing you some relatedness, some differences between the nucleotide sequences, and then kind of some fill in the gaps, like, well, what could have happened here? How do we evolve from here to here? Oh, is there an intermediate sort of step, right? So you're able to do a comparative analysis to figure out if you have any molecular homologies between organisms. So shared bases in nucleotide sequences that are otherwise very dissimilar are called molecular homoplasies. And here's an example here. So you have some that are similar, but overall the nucleotide sequences is otherwise very dissimilar. So it's mostly dissimilar, but you have a couple random similarities. That's not going to be like, oh, the genes are the same. Not exactly. You have some similarities, but for the most part, they're going to be pretty different, because the majority of the sequences are different, which means that they're going to create different mRNA, which means they'll make different proteins, which then have different purposes, structures, all of that. So shared characters are used to construct phylogenetic trees. So that's kind of what we've been talking about here. So once homologous our characters are identified, once you know what makes these organisms similar, you can use them to infer the phylogeny, the evolutionary relatedness diagrams, to show you the lineages over time. So cladistics classifies organisms by common descent. A clade is a group of species that includes the ancestral species and all of its descendants. So it's kind of like the initial node and then anything that's come after that. So it might not be, it's kind of like a smaller portion of a phylogenetic tree, which is typically what we look at on paper because you can only see so much on one sheet, right? Clades can be nested into larger clades, but not all groupings of organisms qualify as clades. It has to include the ancestral species as well. So a valid clade is a monophyletic clade, and it's signifying that it consists of an ancestral species and all of its descendants. So that is a monophyletic, that is one clade. So this is what they look like here. So you have a monophyletic clade, you have a paraphyletic group, and a polyphyletic group. So looking at monophyletic, it has the ancestral species, right? This whole thing would be like a phylogenetic tree, we'll say. So here we have a clade in the blue box, group 1. Okay, you have a ancestral species and all of the descendants. In a paraphyletic group, we're looking at group 2 here in the blue box. You have an ancestral species, but you're missing one of the descendants. Do you see how G should have been included because it is a descendant of this common ancestor? Okay, and then a polyphyletic group is going to be group 3 here. So this is when we have a common ancestor. Perfect. We have A, B, and C. Cool. That's looking good for monophyletic. But then we also include D. And earlier we said just because two taxa are next to each other, C and D are next to each other, does not mean that they are extremely related. Well, of course not, because look, you have all these node points that exist between the two of them. So they're quite distantly related. B and C would be the most related here. Okay. But this has, by polyphyletic, it has the monophyletic group as well as another descendant from a different common ancestor that's still part of the same phylogenetic tree, but it just includes a different branch that doesn't have any connection to the original clade. Okay, the next couple slides go on to explain those. So paraphyletic grouping consists of an ancestral species and some but not all of the descendants. So paraphyletic includes the ancestral species and some but not all of the descendants. Okay, polyphyletic grouping consists of various taxa with different ancestors. Polyphyletic here, different common ancestors, because D does not have a common ancestor with A, B, and C, except for the one all the way back here, which is not included in the clade. Okay. Shared ancestral and shared derived characters. So in comparison to its ancestor, an organism has both shared and different characteristics. That makes sense. Characteristics that have kept the same are shared, or if they are derived, it means that they are newer. Okay, so a shared ancestral character is a character that originated in an ancestor of the taxon. So it came earlier. A shared ancestral came from the ancestor. A shared derived character is an evolutionary novelty unique to a particular clade. So this is a derived character. It's derived independently from the ancestor, okay? So a character can be both ancestral and derived depending on the context. So it just depends on the particular organism. It depends on the particular character and how the phylogenetic tree has been created. Inferring phylogenies using derived characters. So when inferring evolutionary relationships, it is useful to know which clade a shared derived character first appeared. So you need to kind of backtrack beyond that clade and see further in evolutionary history, like if that shared derived character appeared anywhere else. Okay, so this is how you do it with a character table and a phylogenetic tree. Now this character table got 50 shades of messed up because formatting. And it's a PDF. I can't even go through and try to change where the boxes are. But you can kind of see what this is supposed to be. You have the organisms at the top, and you have the characters on the side, right? You have the lancelet, lamprey, bass, frog, turtle, leopard. Those are all across the top. Those are the taxa. Those are the groups of organisms. The characters are either shared ancestral or shared derived. The vertebral column, the hinged jaws, the four walking legs, the amnion, the hair. Those are all of the characteristics. So animals, characteristics. Then in this chart, you have ones and you have zeros. Now notice that the zeros are actually over like the first column, like it's shifted to the left, it needs to be shifted like one box over. So the leopard would really have 1, 1, 1, 1, 1. The turtle would have all ones except for the 0 at the bottom. So it should really be over like one space. Okay. Sorry about the formatting. So you can use this character table to then create a phylogenetic tree. Okay. So looking at the lancelet, now remember the formatting is a little messed up. Really, the lancelet, look, has 0, 0, 0, 0, 0. So the lancelet does not have a vertebral column. It does not have hinged jaws. It does not have 4 legs. It does not have an amnion. It does not have hair. So you have this phylogenetic tree where you have a common ancestor, whatever it was. And the first thing that's the outgroup is going to be the lancelet, because it doesn't have any of these characters that you're starting to study, any of these characteristics, right? The next one, the lamprey, has a 1 and all these zeros. Remember, it's offset because of formatting. So it has a 1 for what? For vertebral column, the backbone. It has a backbone. Does it have a jaw? No. Does it have legs? No. Does it have an amnion? No. Does it have hair? No. So it has one of these characteristics, which is the backbone. So you have the lancelet, which had nothing, and then you have a branch point, and you're going to have something develop here, the vertebral column, because that 1 tells us that it has a backbone. And if you notice, everything else also has a backbone. That's why these little colored boxes are in the background. So 4 out of the 5 organisms, 5 out of the 6 organisms has a vertebral column. So it developed here. You can think about this like time. So this is before the back, 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 before the, oh, we have a backbone. Look at that, backbone right here. So everything that comes after, right, anything after this point right here, I don't have my pen anymore, but the lamprey comes after this point. It is to the right of this point right here. It means it has to have a backbone. Okay, the bass, it has a backbone. A frog, yep, backbone. Turtle, backbone. Leopard, backbone. Because this came first. It's to the left. It's like a timeline. This came first. Everything else that came this way has to have it. Same thing for the hinged jaws. Okay, hinged jaws. That first came in the bass. The bass has the hinged, I'm sorry. Yes, the bass is over here because it's offset because of weird formatting. Right? Hinged jaws developed after the backbone, because it's to the right, so after hinged jaw. Hinged jaw is here. Now, what do we know to be true? If it developed here, does anything before it have a hinged jaw? No. So does the lamprey have a hinged jaw? No, because it came before, right? It broke off here. It broke off before the hinged jaw developed. Does a lancelet have a hinged jaw? No, it does not. Okay, does a bass? Yes. Does a fish? Yes. Or a frog? Yes. The turtle, the leopard, yes. Okay, what's next? 4 walking legs. Does a bass have 4 walking legs? No, because the bass came first. Then the 4 walking legs developed. Does that make sense? So you can just kind of go on, and I mean, you know about a lot of these organisms, like for hair, for instance. You know that a turtle does not have hair, so it came first and then hair. Right? So this is showing you how you can use a character table, even when they're messed up, to kind of guide you to create your own phylogenetic tree. Okay, so an outgroup is a species or group of species that is closely related to the in-group. Okay, so remember, our outgroup is here. In-groups are going to be here. Okay, the various species being studied. So that's everything else. So the outgroup is a group that has diverged before the in-group, okay? So before the in-group. So one of these things is not like the others. The lancelet doesn't have anything, okay? So that's why it is the outgroup. Systematicists compare each in-group species with the outgroup to determine, to differentiate between the shared derived and shared ancestral characteristics. So it's how we're able to choose where to put all of these characteristics that have developed over time. Characters shared by the outgroup and in-group are ancestral characters that predate the divergence of both groups from a common ancestor. So, for instance, if we had something that developed like here, then that means that everything has it. But in this case, all of these are shared derived because they are not present in the ancestor. Because the lancelet is going to be most like this ancestor. and it does not have any of these characteristics. So that's how you read those. Okay, phylogenetic trees with proportional branch lengths. So sometimes the branch lengths matter, but it will always tell you that. So in some trees, the length of the branch can reflect the number of genetic changes that have taken place in a particular DNA sequence in that lineage. So I said earlier that the length of the branch is not equal to the number of changes, is not equal to the number of alterations, descent with modification that's happened there. Sometimes when we're dealing with the ones specifically based off of a DNA sequence, that is true. The length is dependent, but those are usually indicated because for the most part, the length isn't something that we look at unless it is indicated because it's a special kind of phylogenetic tree that is based off of the DNA sequences. Like we've learned previously, some phylogenetic trees are constructed based on different data. If we're using DNA sequence as the data, then sometimes we have the branch lengths that reflect the number of changes that have occurred in that particular lineage. So this would be an example of all of that. So the Drosophila have diverged first. They are the outgroup here, where they have the longest line, whereas the mouse and the human diverged most recently, and they have obviously the shortest line here, okay? So the branch lengths can represent genetic change. So this particular phylogenetic tree was constructed by comparing the sequences of homologs of the genes, which we know if we're dealing with genes, we're talking about DNA, that's going to play a role in development. So the Drosophila was used as an outgroup. Remember that that's going to be different from the in-group. The branch lengths are proportional to the amount of genetic changes in that lineage. And then the varying branch lengths indicate that the gene has evolved to at different rates of these different lineages. Okay. So in other trees, remember different types of trees, branch length can represent chronological time, so actual time, and branching points can be determined from a fossil record. We use our fossil records to kind of fill in like intermediates of what we know to be an existent or an extinct organism. So that's an example here that these branch lengths indicate time. The tree is based on some molecular data. So again, DNA data as the same as in the figure that we just looked at a second ago. But here, each of the branch points are mapped to different dates that you can see down at the bottom. These are the different eras, okay, based on the fossil evidence that we have found. So the branch lengths are proportional to time here. Each one of the lineages, each one of the lines that leads to one of the organisms has the same total length from the base of the tree to the branch tip, indicating that all of the lineages have diverged from a common ancestor for equal amounts of time. So same organisms, different type of representation based on either genetic changes or time in this case. Okay, so we're going to look at maximum parsimony. So this just means the simplest form. That's like the easiest way to say that. Maximum, the most, parsimony, just parsimonious. It is the most simple form. Okay, so systematicists can never be sure of finding the best tree in a large set of data. If you have a whole bunch of organisms, it's very difficult to be like, okay, well, this one goes here and this one goes here, but could they be switched? I don't know, maybe. So you look for all the possibilities and then the simplest explanation. So they narrow possibilities by applying the principle of maximum parsimony. So the most simple explanation, okay? Maximum parsimony assumes that the tree requires the fewest evolutionary events. So that is the simplest form, the fewest events. So appearances of shared derived characters, that's the most likely, because you wouldn't just keep having a pop-up of all these different shared derived characters unless it was actually something more distant. So then it would simplify the whole tree as a total. So computer programs are used to search for the trees that are parsimonious. They are the most simple because they have the fewest evolution events. So here's an example. So there's a whole bunch of different finches, like Darwin was studying, right? That they're all very closely related, but you're not sure exactly, like for these three species, for example, 3 phylogenetic hypotheses, either this one, or this one. You're not really sure which one came first. Okay, you have one of these kind of character tables, kind of, and then you've got some other possibilities here. Like, does this one make sense? Does this one make sense? And then our results, the computer would go through and tell you it's this. Well, then you would use the one that has the fewest evolutionary events. So in order to identify the most parsimonious tree for these finches, you would total all of the base change events. The answer is quite a while. You can conclude that from the first tree is the most simple because it only has 6 events, whereas the others have 7. It's more likely that the fewer number of evolutionary events is the one that actually occurred, right? You're not going to have the same event occurring, but in multiple places. Like 6 makes the most sense. It's the most maximum parsimony. It is the simplest explanation, fewest evolutionary events. That's why you would go with the 6 events as your answer here, according to that rule. Okay, phylogenetic trees as hypotheses. So the best hypotheses for phylogenetic trees fit the most data, morphological data. So the structures, homology, and an analogy, the molecular data, the DNA, the fossil record. Do we have intermediates? Do we know where this came from? Phylogenetic hypotheses are modified when new evidence arises. They're changing all the time because we're able to, have faster computers that are able to sequence more genomes. They're able to find fossils that are now explaining why we don't have intermediates between these other 2 organisms that we know about already. Like, we're finding new things all the time, which means that we'll have to go back and edit these phylogenetic trees. They are the best guess, the best educated guess that we have for evolutionary relatedness based on the information at that given time. So phylogenetic bracketing allows us to predict features of ancestors and their extinct descendants based on the features of closely related living descendants. We rely heavily on the fossil record for this. So for example, phylogenetic bracketing allows us to infer characteristics of dinosaurs based on shared characters in modern birds and crocodiles because we know that they are descendants of dinosaurs, which is still really, really, really cool. So here's an example of the little phylogenetic tree here for birds and their close relatives. Okay, so we're able to kind of make inferences about ancestral organisms based on our existing organisms. So birds and crocodiles share several features. They have 4-chambered hearts. They are song organisms, and they build nests. They brood, right? These characteristics likely evolved from a common ancestor because they have them in common, means that they had to get them from somewhere, so that's their common ancestor, and were shared by all of its descendants, including our dinosaurs. The fossil record supports nest building and brooding in dinosaurs because you can actually see fossilized evidence of these characteristics. So molecular clocks also help to track evolutionary time. So we're going to talk about evolutionary clocks next. So to extend the molecular phylogenies beyond the fossil record, you can take into the assumption how the molecular clocks have changed over time. And this is something that's really interesting, a little bit hard to explain. But molecular clocks are basically, you can track what's happening over time based on kind of DNA evidence. The molecular phylogeny is the DNA. Okay, so molecular clock uses consistent rates of evolution in some genes to estimate the absolute time of evolutionary change. So the number of nucleotide substitutions, so that's an A, T, C, or G, substitution, is when you replace one, in related genes is assumed to be proportional to the time since they last shared a common ancestor. So you can look through to count the nucleotide substitutions and compare that to other organisms and try to figure out when they last shared a common ancestor that way, because it should be kind of like this proportional proportion, for lack of a better word. So molecular clocks are calibrated by plotting the number of genetic changes against the dates of branch points known from the fossil record, because you can use carbon dating and all of that. So individual genes vary on how clock-like they actually function, but you can try to get like a graph overall to kind of determine the molecular clock for different organisms. So this one that we're looking at right here shows you the number of mutations and over millions of years. So you can see the number of mutations is increasing over millions of years, which means that we have evolution happening, okay? So this particular one is for mammals, and it's the number of these accumulated mutations, right? These nucleotide substitutions in 7 different proteins that were studied. It's increased over time in a pretty consistent manner for most mammal species. The 3 green points that you can see there are primate species, so like chimpanzees, things like that, whose proteins appear to have evolved more slowly than the other mammals because they fall below the line. So they're just slower evolution. The divergence time for each data point is based on the fossil record, like what we know to be true. Okay, so we can calibrate the molecular clock for a gene that has a reliable average rate. So this nice plotted points here, right? It kind of creates this average rate of change of evolution by graphing the number of these genetic differences. For example, if you have a nucleotide that has changed, a codon that has changed, an amino acid that is different against the dates of our evolutionary branch points that we know because we've gathered that information from our fossil records. And then those average rates of this genetic change are inferred from these graphs and then can be used to sort of like estimate the dates of these different events that can't be, seen or proven from the fossil record itself. Okay, so differences in clock speed. So some mutations are selectively neutral, which means that they have little to no effect on the fitness. It does not affect whether the organism is fit for that environment or not. It kind of has no change. So neutral mutations should be regular, like a clock. The neutral mutation rate is dependent on how critical a gene's amino acid sequence is to survival. So we have differences in the clock speed depending on the type of mutations that are occurring. Obviously, if it's disadvantageous, those organisms are going to be dying off pretty quickly. But if it is a neutral change, there's really, no immediate effect on the organism's chance of survival, they're a little bit harder to pin down. Potential problems with molecular clocks. Molecular clocks do not run as smoothly as expected if mutations were selectively neutral. Like I said, it's very hard to tell if the mutation wasn't something that was obvious, like a wobble position or something. The amino acid sequence isn't going to change, but the nucleotide sequence will change. And those small changes over time can still be clocked, but it's just not exactly a smooth process. You also have irregularities that result from natural selection, in which some DNA changes are favored over other DNA changes. If a change is advantageous, now you're going to start to see a lot more of that in the coming generations. You have estimates of evolutionary divergences older than the fossil record, that they have a high degree of uncertainty, because if it predates our fossil record is because we can't dig that deep, or those fossils might have been destroyed or whatever, just based on, Earth's construction and the, core being so hot. It's hard to tell if you don't have a known time period to compare it to. And then the use of multiple genes may improve our estimates of these molecular clocks. So applying a molecular clock, dating the origin of HIV. So you can actually, there's molecular clocks that have been created for the occurrence of HIV. So here we have phylogenetic analysis that shows that HIV is descended from viruses that infect chimpanzees and other primates. Makes sense because we are evolutionarily related to them as well. HIV spread to humans more than once. So comparison of HIV samples shows that the virus evolved in a very clock-like way, which is how we can apply a molecular clock to it. Application of a molecular clock to one strain of HIV suggests that the strain spread to humans during the 1930s. So here is this molecular clock chart here for you. It's showing you the years. So it's estimated based on what we know today. You can kind of backtrack that line that we have somewhere between 1920 and 1945-ish, it looks like, as a possible time that the disease jumped to humans. It's showing you here that you have the index of the base changes between HIV gene sequences. That's what is being tracked here versus time. So this is representing the, trying to date the origin of the HIV. This particular strain is called HIV 1M. So the black dots, the little data points that are up there, are based on DNA sequences of an HIV gene in a patient's blood, to patient blood samples. And the dates when these individual HIV gene sequences arose are not certain, because the person can harbor a virus for many, many years before the symptoms occur, because remember that they are lysogenic, and we talked about that in chapter 17, and you can have this in your genes for many, 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 many years, decades even, before you have any symptoms. So it's kind of hard to track when these people got the disease themselves. But just based on the information that's gathered, you can project the gene's rate of change in the 1980s and the 1990s all the way backwards to try to suggest that sometime, like I said, it's a range. They estimate around 1930s is when it jumped into humans, but it could have been as early as looks like 1915 to 1945. But that time range is based on the molecular clock data is roughly when it jumped to humans. So new information continues to revise our understanding of evolutionary history. So recently, systematicists have gained insight into the very deepest branches of the tree of life through analysis of DNA sequence data. So the more we're able to sequence, the more similarities, differences, evolutionary relatedness we're able to discern between different organisms. And it changes what we've known to be true in the past. So from 2 kingdoms to 3 domains. So previously, kingdoms were the largest groups of classification system that we had. So like dear King Philip came over. We did not have the domain. We now have that because through our, sequencing different genomes, we've discovered that there's even a bigger group that we're kind of ignoring here. So early taxonomists classified all species as either plants or animals. And we know today that we have more than plants and animals. What about fungi? Fungi have been around forever, right? There were mushrooms in very early times as well. Like that should have been something that's, it grows out of the ground. It must be a plant. Like that, we know that's not true, right? There's also bacteria or viruses are a gray area because they're not alive. But it's something that we didn't know about back then, right? It's just the evolution of our technology and what we're able to discover has changed evolution as we know it, right? So later, 5 kingdoms were recognized. Monera, which was just for prokaryotes, Protista, Plantae, Fungi, and Animalia. Now, more recently, we have a 3-domain system that has been adopted: bacteria, archaea, and eukarya. So bacteria like eubacteria, they're not ancient bacteria. They are just more common bacteria. Archaea is for archaebacteria that are more ancient. They're more of the extremophiles. You have eukarya, which is going to be all eukaryotic organisms. And the 3-domain system is supported by data from many sequenced genomes because now we know, hey, bacteria are also this thing. It's not just plants and animals. We also have fungi and all this other stuff. Right. And now it's not just Monera for all prokaryotes. We know there's a pretty big difference between like ancient bacteria and more everyday bacteria. There's a huge difference in their genes. So now we need to create something else for them. And we might learn even more that's going to make us change our classification system again in several years. It's just more information we have, the better we can classify organisms and learn more about them. So this is kind of like where we are right now. So common ancestor of all life existed at some point in ancient history. You have domain eukarya, domain archaea, and domain bacteria. So actually, according to this, domain eukarya, which we are in, right, as animals right here, right, we're more closely related to ancient bacteria than we are to common day bacteria. And it's interesting because like animals and land plants is what was thought to be it. But look at all these other branch forms. Like there's all these other little life forms that we didn't know even existed. So it's just like kind of crazy to think in a couple hundred years, what else or how is this going to change, right? The 3-domain system highlights the importance of single-celled organisms in the history of life. The domains bacteria and archaea are single-celled prokaryotes, but now remember that we have 2 different domains for them because they are vastly different types of organisms, although they're still prokaryotic. Only 3 lineages in the domain eukarya are dominated by multicellular kingdom organisms. You have the kingdoms Plantae, Fungi, and Animalia, which were highlighted here for you. Okay, an important role of horizontal gene transfer. So if we talk about vertical gene transfer, that's from parent to offspring, or like in bacteria, because they bud, they don't have sexual reproduction. That would be from the one that split into the 2. That would be vertical. Right? Vertical gene transfer is inherited. Okay, horizontal gene transfer is a little bit different. So the tree of life suggests that eukaryotes and archaea are more closely related to each other than bacteria, which is kind of wild and newer in science. And the tree of life is based largely on rRNA genes, which have evolved very slowly. Okay, so allowing detection of homologies between distantly related organisms. So rRNA is what makes up your ribosomes. which is a conserved character because all prokaryotes and eukaryotes have them, so it's easy to study them that way. Other genes indicate different evolutionary relationships. Of course, this is just where you look in the genomes. There have been substantial interchanges of genes between organisms in different domains. Horizontal gene transfer is the movement of genes from one genome to another. So this is not inheritance. It's moving genes from one organism to another, which we do all the time with genetic engineering now. But horizontal gene transfer occurs by exchange of transposable elements. And a transposable element could be like a plasmid. It could be due to viral infection, which we just learned about, and the fusion of organisms together. So a horizontal gene transfer complicates the efforts to build this tree of life because we're assuming vertical, like descent with modification. We're assuming descent is how we're getting change. But we're also getting change from horizontal gene transfer, which really mucks up the whole situation, right? Like it's a little complicated when you add that in. And it's actually going to change the overall picture, which we'll look at in just a second. So horizontal gene transfer may have been common enough that in early history of life, that it should be better depicted as this sort of tangling web, because it is not just, parents, offspring, parents, offspring, parents, offspring, where you have DNA being exchanged. You have these transposable elements, viruses, plasmids, organisms absorbing other organisms that are actually allowing the exchange of genetic information. So really, this is probably more accurate for what our tree of life truly looks like, because you have these similarities between these different domains of life that there's kind of not an immediate explanation for until you look at horizontal gene transfer, which kind of ruins everything that we've built phylogenetic trees off of. But it's just interesting how they're changing already. And in the next, decades, centuries, what this is really going to look like then. It's going to be a lot messier, because I think that we're going to find more and more similarities between these organisms that we previously thought were not even closely related, right? So that's the whole point of these phylogenetic trees. They are hypotheses. They are educated guesses for how things are related to each other. You've learned about all the different ways that you can construct them, what the different lines mean. Sometimes they mean nothing. Sometimes they mean time. Sometimes they mean genetic changes. You'll typically, if asked about one of those things, be given like a caption of an image or something in order to be like, this is based on molecular similarities between blah, blah, blah. And it'll tell you that you need to pay attention to the lengths of all the little lines for all the clades, right? Thanks for sticking with me here. We have 2 more chapters left of this part of biology. Good luck with all this corona stuff that's happening. I'll catch you in the next one.